The background of this assay is in breast cancer research or in breast cancer. A phenomenal thing happens that patients actually, when they're diagnosed with the disease, many of their tumor actually wouldn't come back after the first treatment, which is the surgery. But at the moment, doctors actually don't have any way of knowing for which patients the tumor wouldn't come back and for which patient it would. So what they do, they provide the chemotherapy for everybody. So what happens here, the patients actually for whom the tumor wouldn't come back, they get unnecessary chemotherapy. And as you know, that chemotherapy itself is a toxic agent. So it's not the tumor which kills the patient, actually it's the chemotherapy that kills the patient. So that is where the assay was needed, and there are a few of them actually out there in the market as well, that segregates the patient into high and low risk group, and the low risk group they can avoid the treatment, and the high risk group you treat them. So this actually offers a fantastic benefit to those patients for whom the chemotherapy was unnecessary. Our assay is a combination of biomarker and it's a panel of biomarker. So this panel actually segregates patients into high and low group, and the hope is that, our preliminary data that we have analyzed, it shows clearly that the low group patients actually the recurrence rate is very low. So saying that you could don't give them chemotherapy. So to tell you more, that we have actually two types of assay. One is a qRT-PCR based assay, that means that it would detect the mRNA from the patient sample, and another one is the protein based assay, which will do immunohistochemistry based assay. We are working on this. The poster actually I'm presenting here deals with the protein based assay. The preliminary data that we're working on currently, and the good results that we have found, it clearly shows that depending on the expression level, high and low, the association with survival is very pronounced in our assay. So currently actually we have found effect of individual markers, because we have like 6 marker panel here, but we're currently working on it to develop and to combine and see what a robust assay we can develop out of this. Originally, the biomarker panel was consist of 11 target. So it's an 11 target biomarker panel. But interestingly, when you develop a protein-based assay for that, so we're currently working on it, but not all target would have a good antibody so that you could develop an assay out of that. So the work is currently ongoing. So far we have got good results out of 6 antibodies, and 3 of them also in the pipeline showing very good data. Out of this 11, 1 or 2 actually we wouldn't find any good antibodies, but that's okay, because as I said, this is a combination, this would make a combination panel of biomarker. So the 6 that we have, it shows a promising result. The effect again is to identify women for whom the tumor wouldn't come back. So that means that after the preliminary diagnosis and the preliminary treatment, which is the surgery, they could go home and live a healthy life and avoid this toxic chemotherapy for them. So this is the main benefit of this assay that we are working on. I can tell you about the challenges in the protein assay particularly. So whenever you are talking about a protein assay, immunohistochemistry based assay, that means that you are talking about an antibody as a reagent. So to find out and to identify the high class, reproducible, highly specific antibody, that is a really challenging thing, because commercial antibodies that we work on, they always do not produce the way it has been portrayed. So it is actually on the end user to make sure that your antibody shows, the validation data shows exactly what it is supposed to do. So this is a major challenge, and in our laboratory, the group leader, Professor William Gallagher, he has been working on this field for quite some time, and he's a leading figure in this. So we actually, in our lab, we have a antibody validation workflow that we have developed. So it's a very robust workflow, very stringent workflow that we have. So we actually buy commercial antibodies and validate it ourselves, and it's kind of a tick box exercise. So if it satisfied us in all different categories, say specificity, reproducibility, in many different aspects, I'm not going to go into the technical detail of it, only then we would take this antibody for the next step and put it into the immunohistochemistry assay. Another important thing that we use, which could be challenging, is that quantification of immunohistochemical signal. So we use image analysis. So in our laboratory, we use actually a very, it's a clinically certified, it has been used in Scandinavia, a few countries already, a image analysis software. So we use that software to quantify the image analysis signal. And the workflow is like, we do immunohistochemistry, do the image analysis, get the quantified data, and then we combine these with the patient data in order to see which markers are related to the survival. In other words, we try to see that, whatever data that we have seen in the quantitative RT-PCR assay, we can mimic that in the protein assay. So that can be challenging sometimes, but we've been working on it, and we've got a very good result for many of these targets. Validation actually is a time-consuming process, and then the most important thing, that which patient set that you are validating. So the way we are working at the moment, so the qRT-PCR assay actually, which is much more ahead than our protein assay. So the qRT-PCR assay, as I mentioned earlier, the company Oncomark, which is Professor Liam Gallagher's company, so they're working on it, and they're expecting that at the end of this year or the beginning of next year, they're going to come out for more clinical tests. But protein assay, I would say it is, say, about a year and a half or 2 years away to go to that level, because we are in a very early stage of validating into different kind of patient data set. So our poster that we're showing here, so we have validated into 2 different tissue microarray set coming from 2 different countries. So the data looks promising, but more work has to be done.